the solution here is, in this case, those scaffolds. And the power of multi-electrowriting is that it allows you to have geometrical feature sizes down to the single cell level. And why is that powerful? Because, as I mentioned before, we usually, whenever we culture cells, on 2D-dimensional plates or even on scaffolds, we drive the functionality mostly from chemical signals. And this is based on this, classical dogma of cell biology that says, all the information is in the DNA, and, the function, which means, we create proteins, goes one way from DNA to, transcription, translation down to the protein formation. And why don't we reverse that, actually? And this is a wild idea, and I have faced a lot of criticism from biologists about that. But we envision to use, multi-electrowriting scaffolds specifically because they give you this huge design space of architectures that allow you to tune cell shapes down to the protein level. And by changing, the shape at the macro scale, but also at the subcellular level, you can induce changes at the DNA and control in a more physical way, the functionality at the end. So, imagine having, a multi-electrowriting printer, and depending on its accuracy and how well it prints the scaffolds, this is how well, and also this translates also to the reproducibility of your results in your cell biology experiments.